Hello. Let us discuss briefly on phenomenon called crossover or crossing over in molecular genetics. What's the definition of the crossing over? It is a mechanism of recombination of genes. So these genes recombined, exchange, part from here coming from there, due to interchange of chromosome. Of course, genes cannot walk just by themselves. They are sitting on the chromosome, and piece from this chromosome coming to another one, interchange, segment, as I said, piece, segment, when at the time of pairing, when the chromosome face each other as pairs. Okay? So this is the crossing over. This is the crossing over. Usually by diagram and to become a bit clear. So in meiosis, you know meiosis, when they are divided into two. So in meiosis, chromosome duplicate during interphase. Here is A. Let us forget about the 46 chromosome in human. Let us take only two chromosomes. So, and we mark it one red and one gray. The red will duplicate, become twice, and that one duplicate as well. And this is interphase of meiosis. Okay. This is age is just, remark. This figure, part A, part B, part C. So A is not a name or part of meiosis, just for the shape. Okay, so as simple as that. This is duplicate. From where duplicate? It can't rain amino acids. It is during the protein synthesis, and you are duplicating your chromosomes. You are duplicated first, become large in quantity twice. It is ready to be split. So this is part of the phase. Then these two, the cell cytoplasm enlarged, and you have this spindle formation, and these two chromosomes paired, facing each other, ready to be divided. It was just one cell. Later on it will be two cells. So first duplicated, then the cytoplasm enlarged, spindle form, central chromosome stick to the spindle through their centromeres. Okay, and later on this will be divided into two daughter cells. Okay, you see part of red coming to the gray, part of the gray coming to the red. You see, this is the recombination. So the genes recombinate, transform from one chromosome to another through movement of segment of chromosome. So the meiosis chromosome duplicated during interphase A, and homologous chromosome exchange genetic information during meiosis, as you see in B. B, this is for the shape. Don't mix it with the terminology of the cell division. It is just shape. So in the B section of this shape, you see this is enlarged, divided, and these exchange. Okay, let us see the last part of the figure. The daughter cell divide again. Those daughter cell, this daughter cell and this daughter cell, they will divide again. Here is their, this is first division, and the daughter will divide. So the one become four cells, and here is the reduction. You see, reduction the number. These are gametes or ova. So divide again in meiosis 2 in this part of the shape C, splitting up sister chromatid to form haploid gamete. Haploid, you remember in the terminology, medical terminology, haploid and diploid. In a human being, diploid 46 chromosome, haploid 23. And do you remember that's very important phenomena in a human being and in an organism in general. They have to divide, then after fertilization they will resume the normal, the ordinary number of chromosomes. 23 from the egg, 23 chromosomes, I mean from the egg, 23 chromosomes in the sperm. In the fertilized egg, you will have 46. Okay. However, here the 4. You see these 4? What you can observe? Can you? This is stay normal, like it is grandfather cell, and this one is normal, while here is abnormal, taking part of, and here is abnormal. So not all the 4 chromatids here, not all of them will be affected, only 2. 2 stay normal and 2 recombined. Okay, I think we will explain it explain this more in the next few slides. Right. Chiasma. Well, there is no red and green chromosomes. This is just a diagram to show you. So we have this pair of chromosome and that pair, and here they are. Sorry, it is not pair. I'm sorry. This is a chromosome and this is a chromosome, and they are pair. There are 2 chromatids. These chromatids, not chromosomes. So they are paired at the spindle while the spindle formed. So part from the green, here they are crossing, and this is called chiasma here. And here they will have some exchange later on. So part of the red going to the green and part of the green going to the red. So crossing over between 2 non-sister chromatid. This is 1 chromosome of 2 chromatid. We call them sister chromatids. And these 2 sister chromatid of the green. So they are not crossing over between the green or between the red themselves. No, the crossing over between non-sisters chromatids. Okay, so this chromosome they can't. It is not observed. I mean, they don't. It is not observed that in 1 chromosome the crossover occur within single chromosome. It is coming from 1 chromosome to another and vice versa. Okay, then you will have this division. Okay, and you will find 2 of them normal, similar to their parents, while 2 chromatid crossed over. And of course everyone will become chromosome. This is gametes, for example, or ova. And this one, if it is in a human, this one have 46 chromosome, and here 46 chromosome, while here they have 23 each. Okay. So in most of organism, recombination of linked gene takes place both in females and males. I said more than one time ova, sperm, so it is occur in both sex. Okay, this chiasmata, هي الكيازما, the plural, the single, and chiasmata is the plural of it, occur in both sexes. However, Drosophila is the fly, house fly, Drosophila never in the male, so it is male. They said rarely or never, so for simplicity said in Drosophila male is immune from this, only the female occur. In silkworm moth it seems to absent in the female. Okay, so in the Drosophila male it's absent in the male, and the silkworm it absent in the females, while most organisms including man, these maybe occur in both sexes, male and female. Okay. And we explained this from the first trans or tetrad stage, only 2. Okay, so cytological observation have established one important fact about crossing over, that the exchange of chromosome segment which cause a combination of linked gene occur at the 4 strand stage. We are talking on the stage, not in the 4 strand all of them. No, it will occur on only 2. And these coming from different chromosomes, from 2 chromosomes, not from the same chromosome, but this stage, at this stage. Okay, process of meiosis. Sorry. All right. So the cross, as I said, only 2 crosses, with 2 other preserved. You have 4, 2 of them normal and 2 abnormal. Genetically, it means that of the 4 chromosomes which emerge from meiosis, only 2 can show crossing over at any one level. Okay. Recombination and crossing over. Recombination is a process which can occur in a variety of situations, but the most popular one is in relation to meiosis. So it occurs somewhere, it occurs in other stages, but it is more popular, more often occur in meiosis. Thus, this is a meiotic process that generates haploid product. Haploid. It is half the number of the chromosome. Like in human being, it is 23. So it is generated 23s, here 23 in each sperm or each ova, whose genotype is different. Why genotype different? Because it got segment from other chromosome, and that segment for sure contain genes, and the genes carry the information. So these 2, you have 4, 2 of them different from the origin. They got information different from the others. So this haploid product, whose genotype is different from the 2 haploid genotype constituting the diploid, forming. They are different from their parents. Look, not parent of the organisms. I'm just saying, from the original cell, the daughter different from the mother cell. The product of recombination is called recombinant. Okay, I'm approaching the 15 minutes. I have to stop. Right, this gentleman is called Robin Holiday, and he talked about the junction, and he decided to name it by his family, Holiday Junction, and he died 2014, not very far. Okay, so this gentleman named this junction cross, name it Holiday Junction, and just in time, before 15 minutes, let me stop here and save it for